This review is meant to help prepare for the Biology Keystone exam. At this point, we are halfway through our reviews. We are on topic 5, which is cellular energy. The material covered in this review will cover questions 12 through 15. So what we have to start with here is the idea of energies and energy levels. Ultimately, all living things need energy, and there are different types of energy. There's light energy, chemical energy, potential energy, all of those different types, and all organisms in some way, shape, or form need energy. Most people think about autotrophs as plants, and they think, oh, these are simply organisms that make their own food. It's more than that. A true idea of an autotroph is some sort of organism, including bacteria, and including some sorts of protists, that are able to create their own source of chemical energy, and typically we think of it as food. And we're really going to be looking at plants through photosynthesis is a common source. Most people are aware of that and understand the basics of the process. We talk about autotrophs. And most people, when they hear the word heterotroph, they think of animals and the fact that animals can't make their own food. And that's completely true. Animals and fungi and some also other forms of organisms such as bacteria and even some protists, they have to get their energy from another source. They simply can't convert it from an inorganic kind of force. We as heterotrophs, we need organic carbon molecules like carbs, lipids for energy. So again, we already talked about these in the first podcast in our biochemistry one. Lipids are our long-term energy storage. It's great to have lipids. If you don't know when your next meal is coming from, or if you have an abundant year, you can store extra material. But more often than not, we as organisms, we like to think on the short term. Do I have enough energy to get me from point A to point B? So that's where we turn to our carbohydrates. And we did talk about each one of these little repeating units. That's a molecule of glucose, and it is bonded together in different ways. This is a new one we haven't talked about yet. This would be ATP. And ATP is kind of instantaneous energy. Yes, we talk about carbohydrates as being our short-term energy, but for us to actually use a carbohydrate, we have to go ahead and break up all those carbon and hydrogen bonds to release the chemical energy stored in those bonds. ATP, however, is much, much easier to use, so we would think of that as instantaneous energy for a cell. So ATP is shown up here. It stands for adenosine triphosphate. So you can see here this nitrogen and carbon group up here. That's adenosine. It's one of those nitrogenous bases, a little bit modified because it's adenosine. It's got the sugar here. We have a 5-carbon sugar. And then ultimately we have triphosphate. So we have 1 phosphate group, 2 phosphate groups, and 3 phosphate groups. Our phosphate groups are very negatively charged, and we can see that here is this active region. And somewhere along your science classes, you've learned the idea that opposites attract and like charges repel. So if I start with 1 negative charge, and that'd be AMP, it's not that bad. I can pop another phosphate on, ADP, adenosine diphosphate. It's a little uncomfortable at this point. And then if I try to shove this 3rd phosphate group on, ATP, adenosine triphosphate, I have 3 negatively charged molecules confined in a rather small space. This is not an ideal situation. It makes this a very unstable molecule. It wants to chew off its arm practically. It'll do whatever it can to dump that phosphate. By dumping that phosphate off, it's able to go to a more stable form and energy is released in the process. You can see right here, I have ADP. It takes energy to pop on that 3rd one because it's so crazy. It's a high energy bond. And conversely, we can pop that bond off to release energy to a cell. So ADP, ATP, think of it as a rechargeable battery. In this case, this battery can be recharged. And then over here, if I actually have ATP, I can use the energy in my battery to carry some sort of other chemical reaction out. ATP is used for all sorts of things. It's going to be used to help your muscles contract. It's used to help you move, to help you breathe. It helps cells function. And we talked about last section in the idea of cellular transport. ATP is the energy molecule that allows active transport to take place. So when ions or other materials are moving against their concentration gradient, it's really ATP snapping off that extra phosphate bond, releasing the energy and allowing a reaction to happen. Oh, an overview of photosynthesis. 2 main processes we're going to be talking about in this podcast are photosynthesis and cellular respiration. Photo referring to light, and synthesis referring to synthesis, creation, making. So the idea here is we are going to be making sugar or making energy out of sunlight. It's a little more complex than that. But ultimately what we're doing is we're using energy, and in this case we are doing a conversion of light energy into chemical energy. We're taking energy from the sun and we're storing it into chemical bonds to make organic compounds. Do we have a formula here? For those of you that have not done chemistry recently or have not done chemistry yet in terms of balancing equations, this may seem a little weird, but to kind of put it in English, I have 6 molecules of carbon dioxide and 6 molecules of water. I need some light energy to get this started and some enzymes to carry out this chemical reaction. They make, there's my yield arrow, C6H12O6, sugar in the form of glucose, and they release oxygen. So it's extremely important that we have plants on this planet because they are responsible, along with other photosynthesizers, for making the oxygen that we as human beings absolutely depend on. Conversely, you can take this equation and you can flip-flop it. So I can look at this stuff being the starting ingredients or the reactants and this stuff being the ending or the products. And this is what we do on a daily basis. We take in sugar and energy and we breathe in oxygen. We go through a bunch of reactions. Now we don't emit light, but we definitely do emit CO2. That's what we breathe out when we exhale. And we also emit water. If you've ever breathed against a glass, that kind of fog that comes up, that's just water from your body. So this is kind of what we're looking at. Photosynthesis and cellular respiration, the two things we're going to be talking about, almost opposite, but not really opposite processes. So with photosynthesis, we're really going to be breaking it into 2 different sections. We're going to have a light reaction section or light dependent reaction. They're basically 2 names. It's the same exact process. And we have light independent reactions. And those have a whole bunch of other names I'm going to mention in a second. Your light dependent reactions are called that because they actually need light energy for the reactions to actually take place in here. It's sometimes also referred to as photolysis, and I'll explain why in a future slide, why photolysis is an appropriate name. And the idea here is we are using light energy, we're starting to get things moving, and we're really trying to set up some hydrogen ions for use at a later stage. Our light independent reactions were previously known as dark reactions, and the reason they were known as that is because they don't need light to occur. However, they don't have to take place in the dark, it's just that they don't need light. They're also known as the Calvin cycle, and they're known as the Calvin cycle because of a scientist that discovered it. And sometimes they're known as carbon fixation. And personally, this is the name that I like the best. For me, it makes the most sense. What's happening is we are fixing CO2, not useful to the plant in that form, going through a cycle round and round and round, and basically adding in hydrogens and oxygens to make it C6H12O6 or glucose. The light dependent reactions, there's a lot of stuff going on here, and for the purpose of the Keystone exam, we're not getting to get all the nitty gritty. But what we want to focus on is, again, we need light. So energy is absorbed from sunlight, energy is absorbed from sunlight here. There are 2 different things called photosystems. They are just places where these reactions are taking place. And what we can see here is sunlight gets absorbed, and what happens is water molecules are broken down. So one of the reasons that the light dependent reactions are called photolysis is because photolysis is light splitting. The light energy is splitting a molecule of water. So you can see here it's splitting into 1/2 O2. Remember, plants create oxygen, so the oxygen is created from the water. Oxygen is not created from the CO2. It's created from the water. And then we also get some hydrogen ions and some electrons, and the hydrogen ions and the electrons are going to be kind of used later on. You can see they're being transported across, and the electrons go through. Again, we're not going to focus too much on this side. Ultimately, we're focusing on this piece. We're going to create some stuff down here that's important for the light independent reactions or carbon fixation. And we're able to get some ATP, not a whole lot, out of here just as a result of hydrogen ions moving from one side to another. But what you really need to know about the light dependent reactions is that you are using light energy to split water and to set up a concentration gradient and get things moving. The light-dependent reactions, again, light-independent reactions are the ones that don't need light energy. So this can take place in the dark or it can take place in the sun. We start with carbon dioxide or CO2. And what happens is it goes through a cycle. And again, we're not going to worry about all the different steps, but we're using some energy, we're using some different compounds. And what we're trying to do is we're trying to go through. It's just showing all this stuff. We're rearranging them, we're changing their shape. And then what happens here is ultimately they get to this ideal shape here. Two of them come together and we get a 6-carbon sugar. That 6-carbon sugar happens to be glucose. Again, it's called a cycle because it goes around and around. We're not worrying about the math. We're not worrying about carbon counting. What you need to know from this is the idea that carbon dioxide gets fixed up with the help of these guys to ultimately make a 6-carbon sugar, which is glucose. So again, not too much for photosynthesis, just the basic idea of it. Cellular respiration. When most people hear respiration, think of breathing. I don't want you to think about breathing for this, because you'll just think about lungs, and not all organisms that go through respiration, they have lungs. Mushrooms go through cellular respiration, and they certainly don't have those types of structures. What you want to think about for cellular respiration is breaking those bonds in glucose, C6, H12, O6, and we use oxygen to help break them down, so that's where the 6O2 comes from. And when you go through all that whole process, you're going to release the energy, and you're going to be storing it in a more convenient form. And that more convenient form is ATP. What happens is we start with a process called glycolysis. So you can see glucose goes through glycolysis, or glucose can go through glycolysis here. And basically, there are alternate pathways it can take. And those alternate pathways really depend on exactly what the environment for the cell is. So just a little bit of glycolysis, glyco referring to sugar or sweet, and lysis referring to split or loosen. So the idea here is to take glucose, which is a 6-carbon molecule, and karate chop it in half to get 2 3-carbon molecules. Those 3-carbon molecules are known as pyruvic acid or pyruvate. What happens here is we actually put in 2 ATP, and we really get out 4 ATP. But since we had to invest those 2, we're really looking at 4 minus those 2. So every time we go through glycolysis, we get 2 ATP. It's really not a lot net gain. But for a microscopic organism like a bacteria, or even something simple like yeast, it may be enough to go through their normal processes. You'll also notice we have this NADH guy down here. And again, that's just another thing that is used and is needed. We're not going to worry too much exactly about its function for this podcast, but NADH is just another player to kind of get this process going. Once you go past glycolysis, and you have those 2 3-carbon molecules of either pyruvate or pyruvic acid, cells have a choice. If they are in a non-oxygen environment, or they are cells that can't tolerate oxygen, then they can go through 2 different types of fermentation, a lactic acid fermentation over here and an alcoholic fermentation over here. This isn't the most ideal thing because you're really not going to get any more ATP out of it. If you are a big hunka organism like us, large, trillions of cells, needs lots of energy, and you can tolerate oxygen, as we can, you are definitely going to want to go through this section down here. You're going to go through the Krebs cycle and the electron transport chain. The combination of those 2 things is going to yield much, much more ATP. But again, if you can't deal with oxygen or you live in an anaerobic, non-oxygen environment, you're going to go through fermentation. And what it does is it kind of sets everything back up. So alcoholic fermentation, for example, we go through glycolysis right here. You can see that I'm using this NAD plus to make NADH. What happens in cells is if they keep doing this process, they're going to run out of NAD plus. So what they can do is they can take these pyruvate molecules and then they can go through alcoholic fermentation, and what they do is they convert it ultimately to alcohol. And what happens is as they convert it to alcohol, they make NAD plus. So it almost allows them to kind of cycle on through so a cell can use as glucose. In addition to making alcohol, they also make carbon dioxide, or CO2. So we take advantage of this as human beings. We have yeast, Saccharomyces cerevisiae, making fluffy little bubbles in bread because they go through alcoholic fermentation. The alcohol burns off in baking. And we utilize this in the wine and beer industry by allowing these organisms to manufacture alcohol. They don't make it to use it. It's just a byproduct for them so that they can get this NAD plus to go back through glycolysis. We also have lactic acid fermentation. And we're looking at lactic acid fermentation here. It's a relatively similar thing. First, we're going to go through glycolysis. And the idea here is we go from pyruvate, we change it up a little bit, we get to lactate. And what we can see again is we are not yielding any extra ATP. We're not making any more. We're just setting up this NAD plus to replenish our items needed for glycolysis. Lactic acid fermentation can be used for manufacturing of yogurt. It can also be used for sauerkraut, kimchi, and a bunch of other food products. In addition, if you exercise rapidly, you get a burn in your muscles. What happens is your muscles are using up and trying to break apart your glucose over and over, and they're using up their NAD plus stores. You can't go right into fat burning immediately or anything like that. So what you'll do is you'll go through lactic acid fermentation to get your NAD plus back. What happens is lactate or lactic acid drops down the pH of your cells and causes that burning, unpleasant situation. Simply by breathing deeply, allowing oxygen to get in because you're not breathing enough when you're exercising, you can go over to the other type of respiration, true cellular respiration, and the muscles will fix their pH. However, this is much more ideal. If you are in an oxygen environment, you want to go through actual true cellular respiration. So you can see we're still starting with glycolysis, but what we're doing is we're going over to the structure here, and this structure is our mitochondria. We talked about that in the Khan Academy podcast on parts of the cell, and the mitochondria is the site of cellular respiration. Here we are. We're talking about cellular respiration. Basically, what happens is all these little folds and these different compartments in the mitochondria allow these different processes to happen. So just like in photosynthesis, there are two parts. There's the light-independent reactions and the light-dependent reactions. Light-dependent reactions needed light, light-independent did not. In true cellular respiration and oxidative respiration, we have two parts. We have a Krebs cycle. And what the Krebs cycle does is it takes that pyruvic acid, that 3-carbon molecule, and goes through a series of breaking it down into CO2. It's almost like the opposite of the Calvin cycle. And then we have a process called the electron transport chain. And the idea behind the electron transport chain is we're gathering H pluses again, hydrogen ions. We create a concentration gradient. We push them through a transport protein, and then that transport protein actually takes that energy and converts ADP into ATP. And then we can use that ATP anywhere else. So what we're looking at here in the Krebs cycle, and again, these diagrams have a lot of information in them. We're just looking at the very basics of it. So we're starting over here. We've gone through glycolysis. We had our glucose up here. We snapped it into a pyruvic acid or pyruvate molecule. Don't focus on anything else, but what you can see here is the CO2 gets split off, going on, another CO2 gets split off, going on, another CO2 gets split off. So what we're basically doing is we're going through a cycle, and again, we have different players, NAD+, NADH, FADH, FADH2. All of these different things are just helping us split off CO2. In addition, we are actually able to get a little bit of ATP from this step. Not a whole lot, but we are now higher than our 2 net gain of ATP. And then ultimately, our last step is the electron transport chain. So what happens in the electron transport chain, I'm just going to flip back to this for a second, we have all these little electron carriers, these NADHs, FADH2 guys, and what they're doing is they take their electrons and they dump them in here. And the idea is hydrogen ions want to go from this area to this area, from a high concentration to a low concentration. But remember, they're ions. They can't travel through the cell membrane by themselves. because they're polar and charged. So they will go through this special transport protein. This transport protein is called ATP synthase. It makes ATP. So what happens is every single time a hydrogen ion is able to go through here, ADP, that's when this is two phosphate groups, and a phosphate line up, and as it shoots through, it actually spins this protein, and those two get kind of pushed together to make ATP. Also what happens is these electrons combine with the hydrogen ions once they get on the other side, and they make our water. So oxygen here is used to catch the electrons to turn into water. It's almost like the opposite of photolysis, where photolysis was splitting water into oxygen. Now we're using oxygen and electrons to make water again. So to sum all this up, and again, we're going very, very basics. If this is something that's interesting to you, in your other biology classes, they go into much more detail, but just kind of basics. We have autotrophs and we have heterotrophs. Autotrophs are the organisms that can create their own energy. Heterotrophs need to consume other energy to survive. We are looking at ATP as our main energy molecule. Yes, we have carbohydrates and we have lipids for energy, but ATP is our quickest energy. And ATP works because of those three phosphate groups, and that addition of that third phosphate makes it a very high energy unstable molecule. If I have ADP, I can pop on a phosphate, like we just saw with the electron transport chain, to form ATP. And I can take ATP and I can use it in chemical reactions, like active transport, and I can use that energy, pop off the phosphate, and go back to ADP. Photosynthesis overall is a process that takes light energy and converts it into chemical energy, such as taking my inorganic molecules, my 6CO2 and my 6H2O with some light energy, turning it into oxygen and, more importantly, glucose, which has a lot of chemical energy stored in it. Basically, both photosynthesis and respiration, they're transforming energy in one way to another. Photosynthesis is light to chemical, glycolysis, electron transport, cellular respiration, both of the fermentation processes. We're rearranging those chemical bonds to get the most energy and the most efficient use of them. When we are going through glycolysis, that's kind of where everybody starts if we're trying to do some sort of respiration. We take glucose, we chop it in half, and we start to make some ATP, a teeny tiny bit, just 2 ATP. If no oxygen is present, then organisms are going to go through one of those fermentation processes. They are not making any more ATP, but what they are able to do is kind of set everything up so they can continue to go through glycolysis to get their ATP. However, if oxygen is present, those organisms can go through the Krebs cycle and the electron transport chain to maximize their ATP yield. In this bottom process, you get about 36 to 38 ATP. With just glycolysis and fermentation, you only get 2. So we're looking at a 16 basically yield. It is 18 time yield. It is much bigger and much better for large organisms such as us to go through this process. We would not survive on glycolysis alone.